Hello my dear amazing learners, welcome back to Freehit. I am Nisha, University Gold Medalist, your biology tutor. So let's continue from the last video where we discussed about the structure of Microsporogenesis, okay, and the structure of Andhra as a whole. Now in today's class we'll be discussing about two important things. One is Microsporogenesis, okay, this is very important. Next we'll discuss about the pollen grains, okay, pollen grains which are the product of microsporogenesis, okay. So we'll discuss about the process and the product and where are the products used, how are the products used. We'll discuss everything in detail, okay. So without any delay, let's get into today's class. Okay. So let me give you an overview of what we'll be discussing in today's class, and then we shall proceed with the lessons. Fine. Okay. So what did it told I'll be teaching? Microsporogenesis. Okay. I always tell you to split it into terms so you can understand it better. Okay. So what does micro stands for? Small, right? What does spore stands for? Spores are structures like this, smaller, rounder. Genesis is formation. So basically what does this translates to? Formation of small spores. What do we call these spores as? Pollen grains. Okay. So pollen grains are the small spores that we are discussing. Okay, they'll be very smaller in size and they'll be dispersed in wind. They'll be of different shape, not only this, but they'll be of different shape. But most commonly they'll have this thing, and they have a lot of ornamentation. What do you mean by ornamentation? Like we wear this earring, nose pin, they'll be having beautiful decorations on the surface. We'll look it into every detail, do not worry. Okay, so yesterday we stopped at stuffs like Microsporogenesis. Remember Microsporogenesis? So in this, in the corner we had these near circular structures which we call Microsporogenesis. What we had inside Microsporogenesis? A homogeneous sporogenous tissue. Okay. What did I tell you? After some time these sporogenous tissues become microspore mother cell. Okay. They become microspore mother cell. Just remember, ploidy is very important. Okay. When I say sporogenous tissue, they are all diploid. Okay. Microspore mother cell, they are also diploid. And this microspore mother cell will undergo the process of meiosis. Remember, wherever meiosis comes, it becomes half. The ploidy becomes half wherever meiosis comes. Okay. So microspore mother cell give rise to pollen grains, and these pollen grains or the microspores are all half ploidy. I mean half into 2n, which is basically n. So what do we call them as? Haploid pollen grains. Am I right? We call them as haploid pollen grains. Okay. Then imagine a microspore mother cell to be a cell like this. Just imagine. And because of meiosis, 4 cells will be formed, right? So first division, second division. Just now we can see 4 cells. But these 4 cells will not immediately split and move away. So they will be sticking together. They will be like 4 guys together, like friends. That structure is known as microspore tetrad. Okay, tetra 4. So microspore tetrad. Then what happens? By the process of hydration. D means removal. Removal of what? Water. Hydration. Removal of water. These guys, these friends, four best friends, they begin to move apart, and thus four pollen grains are formed. Four pollen grains from what? One microspore, right? Are you able to understand? We have microspore tetrad, and then water moves away and leaves the four friends separated, and we form microspore individual. So four pollen grains are formed from one megaspore mother cell. Ploidy of microspore mother cell is 2n, and the ploidy of microspores or pollen grains is n. There are large number of pollen grains present within each microsporangium. So imagine an anther has four. Suppose if a microsporangium has 1 million pollen grains, and they ask a question, how much pollen grains is present in a dithecous anther? So bilobe dithecous anther. When it bilobe dithecous, you should say 2 lobes and 2 theca. So 2 into 2, 4. 4 million anther. Suppose it is a single lobed and bithecous, then 2 microsporangium will be present. 2 into 1 million, 2 million pollen grains will be present. So based on the question, you have to watch out it very carefully, and then you have to answer, right? So this is the very simple idea of microsporogenesis. Okay, yeah. Now, as the anthers develop, the cells of the sporogenous tissue undergo meiotic division. So we just discussed. Okay, they will be undergoing what? Meiotic division. By means of meiotic divisions, they will be forming the microspore tetrads. So we saw the four things staying together. Next, what would be the ploidy? We just discussed. Because of the meiosis, the diploid cells will become haploid. Remember, whenever meiosis is involved, ploidy becomes half. Whenever mitosis is involved, ploidy becomes same. You can also remember it this way. Okay. When I say mitosis, just say equal. When I say meiosis, the ploidy becomes half into ploidy. So you would have already been very much strong in this, but still, just remember, small tricks will help. Mitosis is equal to ploidy cell, meiosis is equal to half the ploidy, right? Next, so as each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, because each of them will become into a microspore mother cell, but that is not a case in case of female. We'll look at. So in case of male only, this is possible. So each sporogenous tissue cell can become microspore mother cell, and which can form microspore tetrad. Each one is a potential. Remember, these alternate terms can be asked in statement questions or assertion reason questions. Okay? Microspore mother cell, pollen mother cell. Microspores, pollens. They are used interchangeably. So you must be very careful while using these words, because they mean the same. So the answer would be the same. Then the process of microspore form a pollen mother cell. You can also call it as microspore mother cell. Through meiosis, by means of meiosis, half the ploidy, that is known as microsporogenesis. Whenever you see microsporogenesis, remember, cut. Okay, small spore formation, right? Small spore formation, which is the pollen grains, that is the male part. Okay, remember this. Next, the microspores are arranged in clusters of 4. Of course, meiosis happens and it divides the cell into 4, and they stay as such as long as they have water. So as long as these guys have water, they'll stick to each other. When they lose water, when they dehydrate, the 4 best friends, the 4 person in the microspore tetrad splits and moves away. Okay, as long as there is water, they'll be sticking together, and if not, they'll go bye-bye. They'll not meet again. Okay, then anthers mature. As anthers mature and dehydrate, the microspores dissociate. Yesterday also we saw the line of dehiscence. Line of dehiscence is typically breakage. When plant components break, when they dry out, when they dry, lose water, when they go dehydration, the wall gets broken and the pollen grains are released into the environment. Okay, that's very important. Now, inside each microsporangium, we have thousands of microspores or pollen grains that are released with the dehiscence of anther. When the anther is dehisced, what will happen? Dehiscence of anther will result in the dispersal of millions of spores or thousands of spores, which varies according to species. It's not the same for all species. Okay, yeah. Okay. Now, can you guess what these guys are? These beautiful little stuffs that I'm showing here on screen. Can you see? What are these? Maybe the picture not so clear, but I'm taking it from your textbook. Just look at this. The shape kind of oval. Look at this kind of globular. How does it look like? Corona? Does it look like coronavirus? Yeah, it does. This also looks like corona a bit. And then here you see some coffee shapes. Coffee beans, does it look like coffee bean? See here, it looks like a coffee bean. And then we have all lot of structure, and here you can see a perfect coronavirus shaped stuff. What are these? Bacteria, virus, fungi? No, they are all pollen grains. So that's what I told. Pollen grains have ornamentation. What is ornamentation? They have lot of designs on their structure. Okay, they have beautiful decorations, maybe spines or maybe flattened or grooves. What do you mean by groove? Just like this, lines. They have different ornamentation. They may have net-like structure on their surface. Remember, like this. Okay, so they have beautiful structures all on their surface. They have these beautiful structures present on their surface. Okay, that's what I call ornamentation. Fine. We'll learn a bit about pollen grains. So pollen grains, let me divide it into two. Now we'll be discussing about what? So pollen grains are basically the male gametophyte. So if you remember from plant kingdom, gametophyte has to be haploid. Okay, so in the flow of Microsporogenesis, microspore mother cell is diploid, microsporangium is diploid, anther is diploid, only the pollen grain is haploid. So it takes the position of male gametophyte. Okay, everything, whenever you refer male gametophyte, make sure you are referring to pollen grains. Okay, pollen grains are usually spherical, but still elongated shape, oval, ellipsoid, every shape is possible. Pollen grain has two layers. Okay, what are the two layers? Outer hard layer, okay, then inner layer. It's not like much hard, it's kind of flexible, you can say. Okay, outer hard layer is made up of okay, pollenin, sporopollenin. Okay, this is known as one of the hardest material known. Hardest. Why hardest? It cannot be destroyed by acid. It cannot be destroyed by alkali. Even no enzymes have been found to date which can, like, break down these guys. So sporopollenin is known as one of the hard substance. Okay, and inner layer is made up of cellulose and pectin. So this is relatively, a soft layer, a flexible layer than that. Okay, just let me draw a diagram. Imagine this is your intine. Just imagine. Okay, let me draw with some other color. Red. No, I don't think red will go. Let me put yellow. I would like to give it an ornamentation. Okay, let's have structure like this. And inside this, whom do we have? Inside this, we have the nucleus, okay, then cytoplasm, we have all these things. But you told me sporopollen is very hard. Sporopollen cannot be destroyed by acid, alkali. It cannot be destroyed by, lot of stuffs. We have told that. But the cell has to come out to establish itself in the next flower. How does it achieve? Here is an interesting thing. This cell is not sealed as you think. They will be, they will be small pores. Okay, there will be small pores. So it is actually like this, and in these regions we'll never find sporopollen, and that's how the inner cell grows out. Okay, this is how the pollen grain grows out. Okay, this is known as the pore. Pollen grains has pores on the surface where sporopollen is not present. Okay, sporopollen is the hardest. There no acid alkali enzymes, even higher temperatures cannot break it. So because of this advantage, they will be well preserved. So this is very important. Pollen grains are well preserved in fossils. Pollen grains preserved well in fossils because it is hardest material. Okay, so it can be preserved, and even it is used in forensic science. Okay, to solve murder mysteries and all those things. Pollen grains has this property of getting stick to the fabrics that you wear. So if you go to a place where the flora, that is the plant growing there, is very unique, those special pollen grains will stick to your body. Okay, and when you come out from the place, move to some other place, with that pollen we can identify you went to that place. Okay, so these are being used in murder investigations, like in forensic science it is being used. Okay, so pollen grains have so many importance. Even it is used as supplement, we'll look into it. Fine. Yeah. So with this, then it is used in forensic. It is also used as supplements because it has lot of nutrients. And I have to tell you one more thing. It is used both by race horses and athletes. Why? Because they give more nutrition for them to work hard. So they are being used by the racehorses are being given these supplements. They are being sold as tablets. Okay, that you can buy and consume. As supplements is basically by means of tablets, all those things, which is available in Western countries. We'll see the images. Okay, and then they can also cause allergies. So pollen grain allergy is very common. Some people might be experienced whenever you go to a new place, you start sneezing so uncontrollably. Why do we sneeze now uncontrollably? Because of these minute pollen particle in the air. They'll disturb your hair, nostrils. They'll enter through nostrils and disturb your mucus layer. And because of that, you'll be getting this coughing, sneezing and all those things. So most of the pollen grains are allergic to humans, but some of them are really good. They doesn't cause any big problem. So because of this, what happened? They'll disturb your bronchi because their size is very small. That will cause bronchial afflictions. Okay, afflictions is basically, damage. They can damage your bronchial layer because of their small size. They can easily go in, and they may even cause asthma. I mean, not because of one-day exposure, but prolonged exposure. That may cause all those things. So whenever you remember, whenever you think about pollen grain, make sure you remember all these points on the screen. Questions can come from any part of this line, any line you can take. Acid, alkali, enzymes, temperaturing, they can ask questions from everywhere. Outer layer made of sporopollen, inner layer made of cellulosopectin, and enzyme alkali. It is susceptible to what? It is not susceptible to what? And all those things, where and all they'll be used? They are used as food in forensic and also as supplements. They can cause allergy, notably asthma. They'll be affecting the bronchial part of your respiratory system. A number of questions can be asked. So just remember. And one more thing. Yeah, I think I've just missed the question. 25 to 50 micrometer. Remember the size? 25 to 50 micrometer. It is a very small. Pollen grains are very small. Okay, so let me change the slide. If you want to take screenshot or have a one final look, just have it. Within 1-2 minutes, 1-2 seconds, we'll get move on. Okay, done? Yeah, we'll go to the next slide. Give me a minute. Right. Okay. Yeah. So this was. Can you see the slides? Okay. This one. Yeah. So this is the slide I was searching for. See here, the pollen grains represent male gametophyte. They are generally spherical, which measures about 25 to 50. The points that I underline are the key point that you must always have that in your mind. They are hard outer sporopollenin, high temperature, strong acid, alkali, no enzyme can degrade it. Exine has prominent aperture, which is known as germ pores. The one I was drawing now, when I was drawing, I just removed the small portions. That is known as the germ pore. So those germ pore are the gaps which is present in that. Exine has a fascinating array of patterns and designs. Okay, I hope you understood this. We discussed a lot, so I don't think we need to spend much time. Yes, this diagram, this is important. See here, this is one pollen grain. You can see the outermost exine and innermost intine. Even this is divided into extextine and intextine, but that is not in your syllabus, we just avoid it. And see here, you have one nucleus and the vacuole which is present. The inner layer has plasma membrane and all those stuffs. And here and there you will be having this germ pore. Okay. Then what happens? This is the first, we saw no, the pollen mother cell that divided in meiosis and they gave 4 pollen grains. And one of the pollen grains, all the event I am describing will happen in all of them. Okay, the pollen grain will undergo division, cell division, and it will form a larger, remember this, these keywords are important, larger vegetative cell, smaller spindle shaped generative cell. Okay, we have a larger vegetative cell for nutrition and a smaller cell mainly involved for the reproductive purpose. So it will carry the genetic material to the ovary and it will fertilize the egg present within the ovule. Okay, so here we have that vegetative cell, generative cell, and this is usually spindle shaped. Spindle is like this. Okay, this undergoes a asymmetrical division, and then this is known as 2 cell stage. We have one vegetative cell one generative cell. Sometimes what happens, this divides and it forms two generative cells. Okay. So in 60% or majority of angiosperm, what happened? One vegetative cell plus one generative cell. This is the stage at which the pollen grains are shed from the anther. But in remaining 40%, what happens? One vegetative cell but two generative cell. So this is a stage at which the pollen grains are shed from the anther itself. Okay. So this one generative cell will convert into two generative cell at stigma or pollen tube usually. Okay. So usually we have one vegetative cell which is larger and one generative cell 60%. What happens? It is just spread in the air. As soon as it reaches the stigma, it will undergo mitosis division and it will form two generative cell, or during the passage in the pollen tube it will undergo the mitosis. But in case of remaining 40%, the complete mitosis division happens when it is present in the anther itself, right? Okay. I hope the diagrams are clear. Be clear with diagrams. Never miss any important point that is being told in the diagram. The inner wall of the pollen grain is called intine. We just saw. It is made up of cellulose and pectin. And of course, the cytoplasm of pollen grain is surrounded by the plasma membrane. We just saw. When the pollen grains is mature, when do you call a pollen grain mature? When it contains two cells. One is known as the vegetative cell. Another guy is known as what? Generative cell. Right? Is that easy? So you have the pollen grain. When it is mature, you have two. Vegetative and generative. It is shed at 2 cell stage in 60%. It is shed at 3 cell stage at the remaining 40%. The vegetative cell is always like what? Bigger, bigger, bigger. Vegetative is bigger, abundant of food reserve and large irregularly shaped nucleus. Okay. You can find a very large cytoplasm, nutrients and an irregular shaped nucleus. See, the generative cell is literally small and it floats in the cytoplasm of the vegetative cell. It is spindle shaped. Okay. So remember, the shape of the generative cell is spindle. The shape of the nucleus of the vegetative cell is irregular. Don't get confused with shapes. Okay. Anther shape is tetragonal. Microsporogenesis shape is circular. Nucleus of vegetative cell is irregular. And the shape of the generative cell is spindle. Okay, that are important. Whenever you find these kind of different shapes, you just write it down. Okay. In over 40%, 60% of angiosperms, pollen grains are shed at 2 celled stage. What are they? One vegetative, one generative. Clear? And in remaining, it is one vegetative plus two generative. Is that clear? I hope there must not be any confusion because we have already learned it. I'm just putting it, summarizing, and then we'll move to the next slide. Yep, pollen grains can cause allergies. Bronchial slide is not changed. Can you see? It's not. Okay, so not a big deal there. There was the same point that we revised will be there. Or give me a second. So pollen grains will be causing allergies and bronchial infections, which may be leading to the respiratory disorders. Ouch. Respiratory disorders, which are like asthma, bronchitis. What is bronchitis? Bronchitis is the inflammation of bronchi. Okay, the bronchi that we have, this trachea. When trachea branches, we have bronchi. So those get inflamed, and then rich in nutrients are obvious. I had the image of carrot grass. Okay, Parthenium you would have seen on the roadside, but you may not be wondering Okay, this is the Parthenium or the carrot grass. Okay, so the pollen grains of Parthenium or carrot grass is a very good allergen. They have caused allergy in so many people. They came to India as a weed. Okay, they was not intended. They came along with the wheat being imported from other countries. Okay, so they came along with that. They were spread in our nation, and they are a common weed that has to be eradicated. Fine, then if the image loads, it will be good to see. I don't know why. Oh, it's coming from the first. Fine. Let me move to the last slide. Oh. Okay, just give me a minute. Yeah. So this is the Parthenium I was telling about. You might have seen it. It is very commonly found on all these stuffs. You can see the Parthenium. Their pollen grain are extremely hazardous. I mean, they can cause allergies. Okay, they can cause allergies. These allergies may lead to asthma. All these are prolonged exposure, not immediately or one day exposure, right? Asthma, then bronchitis. Okay, just remember this is your tracheal tube, and your trachea branches at the 5th thoracic vertebra, and at this point, I mean, these stuffs are known as bronchi. Okay, because of the pollen grains deposit or being hit here, what happened? They start to swell, and your respiratory tube becomes narrow, and it becomes difficult for you to breathe. Very simple. Pollen grain enters, bronchi inflammates, breathing difficult. That's it. Simple. Okay, and then you will be having one more question. You may or you may not be wondering. So these pollen grains, we told the pollen grains will be shed maybe at 3 or 2 celled stage. They will be moving in air or water or on surface. How long can they remain in their functional state? Okay, functional is a common term. We call them as viable state. Viable is that ability to perform the function. What is the function? Fertilization of the egg present within the ovule. How long they can do? This varies from one species to another. From species to species it varies. Okay, so in some species it is quite low, in some other species it's quite high. So from species to species there is a major difference. Okay, there is a major difference in this pollen grain viability. Okay, let me give you a quick example. Let me have some cereals. Okay, in cereals we have 30 minutes of viability. Remember, cereals don't grow one here and other one some 100 miles away, right? What are cereals? Wheat, rice. Okay, they don't grow one rice plant here and other rice plant somewhere around. We usually cultivate it close together. Most of them will be like this, so they can easily pollinate or they can easily achieve their result. So the viability of these cereals, which are commonly wheat, rice, maize, all those stuffs are really 30. And when I say Rosaceae, Solanaceae, they can remain viable for month. Rosaceae, Solanaceae and Leguminaceae. So in Leguminosae, they can stay viable for months. And there are also crop breeding program. Okay, let me write on the top. There are also crop breeding program where the pollen grains are being stored at minus 190 degrees Celsius in liquid nitrogen. For example, a plant has very good properties. Okay, so what they do, they collect the pollen grains from the plant, store it at minus 190 degrees Celsius, and they can use it in the place they want, or after some time, after some years, they can use it. Okay, so in liquid nitrogen, we can store it literally for longer time. So if it is kept as such, wheat, rice, all those things have a viability of 30 minutes. Rosaceae, Leguminosae and Solanaceae, all those things have viability of months. But if you want to extend their viability, make them live a little longer, you can store it at liquid nitrogen at 196 degrees, minus 196 degrees, and that is used in crop breeding. Okay, so pollen grains are very important. Okay, pollen grains are very important part of that. So remember, questions can be easily asked from this. So you have two topics to study, Microsporogenesis and pollen grains. Thank you. Happy learning. See you in the next video. If you have any queries, kindly put it in the comment section. And if you also need the material, better tell me in the comment section so I can post it in the WhatsApp group. Bye.